everyone, and welcome to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the online open community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make this webinar possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. So far, we've held over 25 events in this series. We've had so many amazing speakers. And if you haven't already done so, do check out our archive to watch all of previous talks in the series. You can find information about each event on our website at plantae.org and subscribe to our Plantae YouTube channel to get notifications when new recordings are posted. We have more weekly events lined up through April. Visit plantae.org/education/Plantae Presents to register for them all. Next week, we have a special event that's all about dynamic membranes. If you need to leave this webinar early, know that a recording will be made available within a few days. Today's talks will each be 20 minutes, followed by 10 minutes for question and answer. As you have questions for our panelists, please put those in the Zoom question and answer Q&A section. Questions specifically for our panelists can be shared in the Q&A section to make them easier for our moderator to navigate. You'll also notice that there's a chat area open. This section is for you to introduce yourselves, say where you're from, and connect with your colleagues. If you're having technical issues, that's also a good place to let us know about those, because then others can see them and let us know if they're having similar issues. All right. Today's talk is moderated by Geeta Yadav. Geeta is a lecturer at the Department of Plant Sciences, University of Cambridge, and scientist at the National Institute of Plant Genome Research, New Delhi. She is an expert in genomics and structural bioinformatics with applications in food security and phytochemistry. She is part of the Plantae Presents Steering Committee. Thank you for joining today, Geeta. I will hand it over to you now.
Thank you, Katie. And a big hello to everyone here. Good morning, good afternoon, and good evening, depending on which time zone on the planet you're tuning in from. Once again, thank you for checking in to the Plantae seminars and today's webinar. As Katie already mentioned, this is the series going on since last year, and I encourage you to please look at our past sessions, events, and the speakers who've been here with us. Today, we have two stellar plant scientists who are going to be talking to us, both of them brilliant orators and ace biologists, both of them from India. And how often does it happen that two of your favorite speakers, both of whom you forever looked up at, are speaking at the same platform on the same day, and you get to moderate the session? So as you might have guessed, I'm in a good place today, and I'm a fan of both of our speakers today. It's a stroke of luck that I'm on the panel too, here with you. I would like to begin by introducing Professor Usha Vijayraghavan to our listeners first. Usha Vijayraghavan obtained her Ph.D. in 1989 from Caltech, Pasadena, USA. She was working on yeast genetics, and she did her post-doctoral research again at Caltech with Elliot Meyerowitz, none other. And there she began to work on transcription factors, and especially transcription factors that were regulating plant development. Usha returned to India in 1990 when she joined the prestigious Indian Institute of Science, Bangalore. And over the next 3 years, that is between 1990 and 1993, she made it a point to remain connected with Caltech as a visiting fellow on annual 3-monthly academic visits, back to Caltech, where this was the time when she was setting up her own lab at the IISc, and she was also transitioning to rice developmental biology. And in my view, I think this is a really great piece of advice for budding ECRs who are working abroad and planning to return to their own countries for pursuing a long-time career. And if you're not sure whether or when to make the move, think about how Usha managed this transition. Coming back to her, today, Professor Usha Vijayraghavan is at the Department still at the Department of Molecular and Cellular Biology at IISc, but she's also a dean at the Division of Biological Sciences at the Indian Institute of Science. She's now involved in recruitments, promotion, funding, what have you. Her group, she also does, of course, research and teaching. And for research, her group uses genetic and genomic tools to investigate key, factors that control complex gene networks. A focused research area of her group these days is on the regulation of flowering and plant architecture in rice. Today, Professor Vijayraghavan will talk to us about CODE's rice floret development, leads from studies on the OSMADS1 regulated gene networks. Over to you, Usha.
Thank you so much, Gitanjali, and absolutely almost undeserving, flattering comments. But in any event, it's always my pleasure to speak on our work. I first must say how happy I am to be here. And I want to thank the ASPB community, the Plantae steering committee, Mary, Katie, and Gita for organizing today's session. And this webinar really keeps us connected and keeps our spirits high for plant biology right through the pandemic time. So I appreciate the opportunity to give you a glimpse of the work that we're doing. Flowering in rice is marked by the transition of the shoot apical meristem that's established in the embryo from being a dome-shaped structure to something that rapidly elongates, generates axillary meristems on bracts, and these axillary meristems go on to form the primary rachis branches. After making about 6 to 10 branches, depending upon the variety that you're working with, the apical meristem aborts. These primary branches elongate and go on to make at their tip what is a spikelet meristem, and the spikelet meristem is the last final branch in the inflorescence made by rice plants. The primary branch meristem can often give second order branches, which are called secondary branch meristems, and these also can go on to terminate in the final branch meristem, which is called as the spikelet meristem. So clearly the decision to flower involves moving from a multipotent state of the meristem slowly through various developmental states in order to reach something which will now form a determinate floret meristem. So the floret meristem is made on the spikelet meristem, and in the case of rice, a single spikelet makes a single floret, and I'll give you a little bit more about this in the next slide. The spikelet organs are a set of small bracts, which are called rudimentary glooms, followed by 2 slightly larger bracts called as the sterile lemmas. And then come the floret organs, which are the lemma, the palea, and 2 asymmetrically positioned, very small organs, fleshy organs called lodicules, internal to which are present the stamens and the carpel. One must appreciate that this is a drawing of the rice flower after it has been opened. And just as rice, all other grasses have predominantly closed florets, which whose opening and closing is regulated. And I'll tell you a little bit about it in some time. So organs of the rice flower which are unique are the lemma, palea, and the lodicules. And those of the spikelet are these rudimentary glooms that are formed. Let's go a little deeper into looking into what are these organs and what type of cell types do they have. Like I told you, the lemma and the palea are the first 2 organs which are made on the floral meristem. They arise in successive positions. They're oppositely placed, so they're not in a world position. The lemma is larger than the palea, and they are closed in order to give you a section will show you the closed floret, in order to envelop the inner organs and protect them through their development and protect them until they're ready for fertilization. How is this floret kept closed? The floret is kept closed by this specialized structure in the palea that is called the marginal tissue of the palea that hooks with the lemma in order to keep the flower closed. This hooking is temporarily opened when the anthers have to come out of the flower, and during this process pollination takes place, and the exerted anther will allow for cross-pollination. Not only are the organs lemma and palea unique in their functions, closing the flower and protecting them, they also have very distinctive structures Their epidermis has large cuticular deposits, unlike that which is found in the glume or in that of the leaf. And their homology to dicot sepals, which are by analogy the ones that enclose the developing flower, is still debated, right? So iconic work, the iconic ABCDE model by genetic work done in Arabidopsis and other eudicot plants has given us the concept that there are combinatorial action of MADS box transcription factors that patterns the organs and the whorls of the flower. Class A factors together with class E factors give us sepal. A, E, and B give us petals. B, C, and E give us stamens. And C, D, and E give us ovules, which are embedded within the carpel. Right? This framework has been fantastically predictive in many, many plant species. And not surprisingly, the orthologs of these genes are also found in the case of rice. And the rice genome gives us several examples where gene duplication and diversification has contributed to the subfunctionalization of members of these ABCDE class of genes. For example, the class C genes are represented by 2 genes which have subfunctionalized. The pistillata orthologs are represented by 2 genes which are again show some degree of subfunctionalization. In today's talk, I'll be talking mostly about this set of genes, which are the class E genes in Arabidopsis, whose representatives are 5 genes, and let's look a little deeper into them. In addition to using the classical ABCDE ortholog genes for developing the rice floret, there have been new recruitments or innovations by bringing in other factors like DL, which is a YABI domain transcription factor, and bringing in sister factors from the AGL, that is the AGL6 family members, which together with the canonical ABCDE MADS box factors control the formation of the floret, and some of them contribute to the spikelet organs. When one looks at the phylogenetic positioning of rice class E factors, we see that there is a subclade which is called the Lofseb subclade, where 3 members of the class E gene family of rice are distantly related to sepalata 4 of Arabidopsis. Very, very interestingly, the Lofseb clade of MADS box transcription factors have hugely expanded in grasses, with members being present in almost all the cereals and grass family members. This gives an early clue to the fact that these genes might be involved in the innervation of the floral organs that are seen in grass flowers. Another important point to remember is that in Arabidopsis, there are 4 class E sep factors, but these are largely redundant and can substitute for each other's function. And therefore, a complete loss of class E activity is obtained only in the quadruple mutant. Contrast that with the phenotype of mutants in 2 different Lofseb gene members. The phenotypes are very different, indicating that these Lofseb gene members during their evolutionary trajectory have gone on to adopt certain unique developmental roles, while they might share some other developmental roles. OSMADS1 mutants, which are either created by TOS17 insertion, a transposon insertion, a semi-dominant natural allele, or by RNA interference, tell us that MADS1 has a unique role in specifying the floral meristem to be determinate. In the mutants, regardless of what type of allele it is, you have an overproliferation of the meristem organs, and all organ fate is disrupted. This is distinctly different than the mutant phenotype seen for MADS34, its closely related cousin in the Lofseb clade, where the floret organs are relatively normal. And what is defective is that the rudimentary glumes in the sterile lemma, which are the spikelet organs, are malformed, and panicle branching is affected. So 2 closely related members of the Lofseb clade themselves have unique contributions to the development of the rice flower. This allows us to understand how they do so and what are their downstream pathways. The first clues to what OSMADS1 may be doing come from a combination of our work done to map its domain of expression during floral development, and understanding what are the pathways which are deregulated in the on the knockdown of MADS1. MADS1's expression starts in the incipient floral meristem just after the spikelet sterile blooms have been specified. Its expression is uniform, but as we go through development, its expression is spatially controlled and restricted to the lemma and the palea with a tiny bit of expression in the carpel. In mature flowers or near mature flowers where organ differentiation is going on, its expression is restricted to the lemma and the palea. When you lose the expression of MADS1 or you greatly reduce the expression of MADS1, we find that up to about 15% of its downstream genes are other transcription factors. And about 20% of its downstream genes encode for factors which are involved in cell signaling, yet others are involved in cell cycle, and a large proportion of the downstream pathways to MADS1 are involved in metabolism and catalysis. And metabolism, as all of you might appreciate in plants, is very closely linked to the production of hormones and signaling. So in order to understand if there is a large swathe of 6,000 odd genes whose expression goes awry in the reduction of MADS1, what of these are direct effects and which of these are domino effects? In order to probe that, we decided to raise antisera against MADS1 and use these antisera to do chromatin immunoprecipitation and prepare chromatin from panicles which are at very early stages of development. So these are panicles which are specifying flowers. That is, they have flowers of this range to this range. So that's about 2 millimeter to about 2 centimeter. And Illumina sequencing tells us that there are target genes to which OSMADS1 is bound. And those binding sites which are associated with genes contribute to its direct targets. And this is around 3,500 direct targets. As expected for a transcription factor, it binds around the transcription start site of its target gene. It also binds upstream, and surprisingly, up to 50% of its binding sites lie within the gene, either in exons or introns. This suggests that MADS1 binding sites potentially are enhancer elements controlling the expression of the gene that they are associated with. About 64% of the binding sites have the typical CARG motif, which is the motif bound by the MADS DNA domain, MADS protein domain that binds DNA or its variants. But surprisingly, this very deep analysis of the binding sites of MADS1 on the rice panicle genome told us that CARG motif is not a necessary feature of the sequences to which it's bound. OSMADS1 is able to bind sequences where the majority of the binding sites are just simply A tracks, that is the core of this element without the CG flanks. And another feature that characterizes the MADS1 binding site is that surrounding the binding site, the DNA tends to be unstable. So the binding of MADS1 to its target loci is probably a combination of the presence of the cis element, but also the biophysical characteristics of the DNA. In order to get a true picture of the real downstream effects of MADS1, one needed to have a null mutant. And most of the mutants that I've described to you before were either partial loss of function or knockdowns or transposon insertions and semi-dominant alleles, etc. So we generated a clean null by doing a deletion disruption by homologous recombination with our collaborators, Professor Veluthambi at Madurai Kamaraj University. And the null phenotype told us, reaffirmed that MADS1 is really a very, very early acting upstream regulator of floral meristem identity. In the absence of MADS1 or in the knockout, all floral organs become leafy or bract-like, stamen number is reduced, lodicules become fused to stamens, stamens get kind of like sterile lemmas, and there is a mass of carpeloid tissue which arises, and these two are mosaics. In very extreme cases, we see that the floral meristem not only loses its determinacy, keeps on proliferating, but it also reverts to an inflorescence state. So here is a single floret which has transformed into an inflorescence branch, generating many lateral florets, and all the lateral floret organs are leaf-like. So MADS1 is therefore required not only to establish a floret meristem, it's apparently required to maintain the floral meristem characteristic. In order to quickly understand, are genes which have been implicated by other studies to be regulators of MADS1, affected in our mutant, we cherry-picked those genes which are well characterized by means of mutant studies. For example, MADS34 is its cousin. And MADS34 expression in very early florets was high, whereas in late florets, which are lacking MADS1, the expression level was low. Does this match with the phenotype? Yes, we think that the high level of MADS34 in MADS1 knockout mutant persists the meristem in a spikelet state and prevents the appropriate transition to the floral meristem state. Another floral meristem regulator, which is well characterized, is MADS15, which is also called full one, or belongs to the AP1 clade. And MADS1, together with MADS15, are known to be required to establish the floral meristem. So in MADS1 knockout, the genetic interactor and partner of MADS1 is also downregulated. So obviously these two are in a feedback loop which maintain their expression and maintain the identity of this floral meristem as being floral. We found that two genes, which are, one is a class C gene and the other is a class D gene, their expression levels are highly reduced, and these together could explain why the floral meristem becomes indeterminate. So it behaves like a class CD minus in this case. Certain genes by mutant studies have been shown to be involved in lemma and palea development. And shown here are three such genes, SPL16, DH1, and DP1, of which DP1 mutants are defective only for palea development. All of these three genes we find are much affected later in development, with early developmental effects being marginal. So genes such as MADS34, MADS15, MADS13, 58, and these organ differentiation genes confirm that MADS1 is a regulator of many processes through the formation of the floral meristem and floral organs. We had now a beautiful mutant, which became a wonderful tool for us to understand the transcriptome landscape that is changing as development proceeds. So we binned mutant panicles into two stages: early stages when the flowers are at the early meristem stage or when organogenesis is just beginning. And we binned panicles where flowers are in the differentiation stage somewhere here, determined the transcriptome, took the differential transcriptome and overlaid it with the direct targets of MADS1 that we had determined by ChIP-seq. This analysis allows us to define that in early floral meristems, about 570 genes are directly regulated by MADS1, and the domino effect of this direct regulation is the expression levels of about 3,000 to 4,000 other genes are affected. So the very observation that MADS1 can positively regulate genes as well as negatively regulate genes indicates that MADS1 must partner with other factors which can change the chromatin status to bring about either activation of gene expression and transcription or repression. These are questions that we're addressing in our ongoing studies. Surprisingly, by binning these developmental flowers into two developmental bins, we're able to say that MADS1 is not only required early in development to establish the floral meristem and the organ primordia, it continues to be required later in development when differentiation is going on, because 166 genes are bound by MADS1 and their expression is affected later in development. Now we passed the genes which are affected directly and those which are affected indirectly into different pathways. And we're able to now understand that pathways like hormone metabolism, genes involved in hormone metabolism, transcription, and protein are directly affected early in development, but they're also affected later in development. But certain pathways are predominantly affected in either before or later. For example, sulfur assimilation is something that's affected later, whereas secondary metabolites are affected later, whereas primary carbohydrate metabolism is repressed in early floral development. So I'll just give you a quick snapshot of what MADS1 is doing to other MADS box factors. So MADS1 regulates many transcription factors and chromatin modifiers, including AP2 EREF domain containing factors, MIB factors, Jumonjis, HATs, and so on. Since we know a lot more about floral MADS, I'm just going to show you what it does with respect to MADS box factors. So OSMADS, in the absence of OSMADS1, we see the expression level of many of the other floral classes, other classes of floral factors, A, B, C, and E, is downregulated. That means MADS1 is actively promoting the expression of these genes, but these genes are not direct targets of MADS1. So therefore, we expect there is an intermediary by which this expression level of these other floral factors is maintained. This contrasts with the fact that in Arabidopsis, SEP3, which is its very, very distant related MADS box factor, SEP3 directly regulates ABCDE class of factors positively. So while MADS1 and SEP3 are distantly related, the wiring between MADS1 and its downstream MADS factors and the wiring between SEP3 and its downstream MADS factors has slightly changed. There's an indirect relationship here, whereas there is a direct relationship over here. We took all the direct targets of MADS1 in early development and tried to understand how we might bin them into different functional groups based on what is known about the properties of their predicted downstream targets. By this biotapestry plot, we're able to present to you a set of genes which are predicted to be involved in meristem maintenance, or some of them mutants are available. For example, genes involved in gibberellic acid biosynthesis, and very, very promisingly, two factors, OSH15 and OSH1, which are stemness factors. These are the relatives of STM and Arabidopsis, and the Cli peptide gene, OSBAM2, which is sort of working in a parallel pathway to clavata3. Seeing genes like these appearing as direct targets of MADS1 indicates to us how early in floral development MADS1 might be maintaining the meristem and creating more cells in order for organ differentiation programs to initiate. We're also able to map sets of genes which are involved either in control of cell shape or in the control of meiosis or in the control of stamen development. And these set of genes give us promising leads on how one might perturb sub-pathways within the big field of, within the broad pathway of floral formation, floral organ formation. We have similarly candidates which are predicted to be involved in carpal development. I want to give you a very quick glimpse of a little bit more that we have understood by our meta-analysis of maintenance of the meristem. So I've shown you here that MADS1 directly regulates positively OSH15 and OSH1, which are homeodomain transcription factors. And these factors are the relatives of STM1, and they are required to maintain the floral meristem early when it is being established. So MADS1 sets up a positive feedback loop with these stemness factors, maintains this meristem, and in collaboration with them controls 53 other factors, which are either transcription factors or chromatin modifiers. So this network has been built by a meta-analysis of our data on MADS1 combined with the chip sequencing data, which has been done by the group of Suda et al., where they have mapped the direct targets of OSH1 and OSH15. Remarkably, because we have the data sets from Arabidopsis on what are the direct genes and targets bound by Arabidopsis SEP3, we are able to understand that many of the gene targets controlled by this feed-forward loop in rice are probably unique to rice. Many of the direct effects of this feed-forward network on these 53 other factors, which are unfilled circles, are targets which are regulated in the case of rice, but their orthologs are not regulated by Arabidopsis SEP3. So this gives us exciting glimpses on the evolution of genetic networks, of gene regulatory networks during the development of flowers. So where do we go from here on? We really are scratching the surface of this very complicated network, which gives you a very precise outcome. And what is the precise outcome? The outcome is a determinate single flower with a fixed set of organs on the floral meristem. We need to understand, while MADS1 might be an early player in this game of making a floral meristem and all the floral organs, how does it do so? What are its partners, and how do they together control these developmental outcomes? Secondly, once a developmental pathway is chosen and established, how is it maintained and how is this network fine-tuned? We predict that since MADS1 is directly controlling the effect of many epigenetic factors, including factors like Jubon Gs and histone demethylases, histone acetyltransferases, as well as trithorax group of proteins, it's probably maintaining the expression status of its downstream genes by co-opting chromatin modifiers. So our future studies, we are well into them, are looking at a couple of partners of MADS1, a couple of targets of MADS1, and some epigenetic modifiers, and how do they all tie up to take this apparently messy network of developmental gene expression to give you a predicted outcome. All of this work done over a couple of decades has been possible only because of a fantastic team of students and postdocs that I have been blessed to have over the couple decades. Many of my students and postdocs are now themselves professors, and you probably will hear from them in this webinar series in some time. Many collaborators who've been very generous with their time and reagents, and listed over here are some of my more recent collaborators. Funding from various funding bodies in the institute, within the institute and within Government of India. And last but not the least, the Rockefeller Foundation, whose traveling fellowship allowed me to be connected with Caltech in the early days when rice developmental biologists were really very, very few. You could count them on the digits of one hand. And that was a time when I was scratching my teeth or cutting my teeth on developmental biology and rice. It's an exciting time, and it's always been a pleasure talking to people and learning more. I look forward to your feedback, and thanks for listening.
Great. Thank you so much, Usha, for that insightful lecture on ERISA master co-regulatory networks during development. I'm so glad that you talked today about, you didn't stop at looking at the differentially regulated genes. You moved on from there by filtering these thousands of genes by direct or indirect interactors, by doing ChIP-seq. So you've overlaid ChIP-seq with RNA-seq, and then you go on further from there to find the master regulatory network of how MADS1 is looking at other, controlling or regulating other transcription factors. This is exactly what I'm so glad, because today morning I was taking a class with PhD students here at an IPGR, and I was trying to tell them that you're just scratching the surface when you're only looking at the differentially regulated genes. There's so much information there that we can go for, and your talk has been perfect there as an example today. I hope some of our students have been listening. So thank you for this talk, and you've been perfectly in time. And we are now open for questions. I do have a couple of questions in the chat box. What I request is, if there are more questions, please, just a message to our listeners to please add your name along with the question so that I can ask it on your behalf. So the first question, Usha, to you is from Hasti Ram, and he's asking, you had 3,000 MADS1 targets, but then you only found altered gene expression for just 200 of them. How would you explain this drastic change in number?
So Hasti, you have to understand that we've taken a snapshot of development. So if you look at the ChIP-seq data sets, we binned everything from early flowers to flowers which are undergoing organ differentiation. So that gives you the overall data sets of genes where MADS1 is bound. But binding doesn't mean change in gene expression. So the 200 or 500 genes that I told you whose expression was changed in a particular developmental stage, in another developmental stage, it might be an overlapping set of another 200. At another developmental stage, it could be another overlapping. And collectively, this could be explaining a larger number of the binding sites. It's not surprising, both from plant work as well as from animal work like in Drosophila, that in many times transcription factors are bound, but that doesn't mean that they're acting at that time and point. Many of the times it depends upon whether they receive a signal, whether their partner is present, whether the tissue is competent to respond to the signal, and so on. So binding does not ensure that your expression is occurring at the same time and place. So we have 200 genes in that particular window, but in another developmental window, we might find yet more and more and more genes.
Thank you, Usha. And I hope that answers your question, Hasti. There are two more questions from Isha Joshi, and I'm going to combine them together for you, Usha. She's asking, in your, did you find the MADS1 binding site in any cell cycle regulator genes after doing the check? Also core histones, she's very interested. Would you like to?
Very, very, very interestingly, yes, we did. I mean, these are, as you might have noticed, the later part of the talk is a paper that we are writing up. So it's unpublished. Yes, we do find cell cycle regulators. So these, remember now the cell cycle regulator could be either directly or indirectly controlled. So we have an instance where it is directly controlled, and we have another instance where a cell cycle regulator is indirectly controlled.
All right, Usha, there's one last question again from Isha. She's asking, how did you cut short to the trithorax group of proteins out of all the chromatin modifiers that you were looking at? Did you see any interaction there, or was there something else that focused you onto these trithorax?
This is ongoing work. See, like I told you, there are several chromatin modifiers which are affected. Okay. So we see that, for example, there is Jumonji. There's a set of Jumonji factors which are affected. There's a set of histone deacetylases which are affected. But I kind of put a preference on the trithorax for functional characterization because we had already done a study with the polycomb group of factors. So together with our collaborator, Sundareshan, who is at UC Davis, we had shown that EMF2B, which is one of the members of the PRC2 complex, actually is involved in developmental regulation of the class E factors. So EMF2B is controlling floral organ identity through setting the chromatin landscape of class E factors. And included in these class E factors was MADS1, but it was doing so indirectly. It was not doing so directly. So having looked at the PRC2 end of the story, we wanted to see how is it that trithorax end of the story might be working, because the trithorax end of the story, a trithorax group of factors are principally involved in setting up activation marks in their downstream targets. And we found that one of the trithorax group of proteins was, the gene that encoded it was directly controlled by MADS1.
Okay, so does that, Isha, if that answers your queries, please type back in the chat box. And if you have any further queries, don't don't stop. Please do mention them, and I will ask them on your behalf. Even if after Dr. Sonti's talk, we will again be having a question session. If there are any more further questions for Usha, please feel free to type them out. Once again, thank you very much, Usha, for your talk.
Thank you. I wasn't watching the time. I hope I didn't keep Ramesh waiting too long.
No, not at all. The timing was just right. In fact, we've got 20 minutes. You were 1 minute up and down here. Dr. Sonti, I have your slides with me, but I think
Great.
You're perfectly, you're totally all right with that. So I'm going to just move on now and introduce Dr. Ramesh Sonti, after which he can check if his slides are working. Otherwise, don't worry, I've got it handled here. So Dr. Ramesh Sonti obtained his bachelor's degree in life sciences, botany, zoology, and chemistry from Andhra University in Vizag, India. He obtained his master's and MPhil degrees in plant sciences from the University of Hyderabad, and then he got a Ph.D. in bacterial genetics from the University of Utah. This was followed by a postdoctoral stint in plant genetics at MIT. And in 1993, he returned to India to join the CSIR Center for Cellular and Molecular Biology, CCMB, again at Hyderabad. In November 2017, he came here to Delhi to join us at the National Institute of Plant Genome Research as our director. And just last year, he served as director at NIPGR only till, just till last year when he superannuated in May 2020. Currently, Dr. Sonti is a JC Bose Fellow at the CCMB Hyderabad, and his research interests are in the area of plant microbe interactions, development of disease-resistant rice varieties. He's also a recipient of a number of awards and fellowships, including the prestigious Shanti Swarup Bhatnagar Prize in Biological Sciences, which is one of India's absolutely the highest science awards. He's also a fellow of the Indian National Science Academy, New Delhi, and the National Academy of Agriculture Sciences at New Delhi. Dr. Ramesh Sonti will be talking to us today about plant innate immunity, induction, and suppression of host innate immunity in rice-Xanthomonas interactions. Over to you, sir.
Ramesh, we can see your slides, but you're muted.
Okay.
There we go. Perfect.
Yeah, great. Thank you. Thank you, Gitanjali, for the nice introduction, and thanks to Mary and Katie for organizing this session, and thanks to all others whom I can't see, but thanks. Some of you have probably woken up very early in the morning. Others are probably late into the night. And thanks to all of you. See, my lab has been interested in understanding how plants sense the presence of a pathogen, how they mount an immune response, and how pathogens actually suppress this immune response to be able to cause disease. And the model system that we use is the interaction between the rice plant and a bacterial pathogen called Xanthomonas oryzae pathover oryzae. It's a big name, but I'll just call it XOO for short. The immune responses that we study are those that are induced in rice upon damage to the cell wall that is caused by enzymes secreted by XOO. Now, this is a story that started almost 20 years ago when a PhD student in my lab found out that this XOO bacterium secretes a variety of cell wall degrading enzymes. These enzymes target different components of the rice cell wall. There are cellulases that target cellulose, xylenases that target xylem, pectinases that target pectin, and a variety of other such enzymes. And what the student showed is that mutations in genes for these cell wall degrading enzymes affect virulence of XOO bacterium. And if you make 2 mutations in these enzymes, the virulence deficiency becomes even greater. So for each of these enzymes, or for many of these enzymes, he could show that there is a measurable effect on virulence upon mutating them. So these enzymes are very important for XOO virulence on rice. Now what we realized was that in the 80s, Professor Peter Albersheim, a very famous American biochemist, showed that cell wall degrading enzymes secreted by a fungal pathogen trigger immune responses in soybean cells as a result of damage that is caused to the soybean cells by pectinase action. He was also able to show that it was oligogalacturonides, that is pectin degradation fragments that are released upon action of pectinase on soybean cells that are triggers for the immune responses. So the student, who is actually Gopalji Jha, a faculty member at NIPGR, the institute where Gitanjali works, what Gopalji did was, his doubt was, if the cell wall degrading enzymes in a fungal pathogen of soybean can elicit immune responses, would the cell wall degrading enzyme secreted by XOO also induce similar immune responses in rice? And this was relatively straightforward for us because we had already got many of these cell wall degrading enzymes purified and they were at hand. So what Gopal did was treated rice cells with the cell wall degrading enzymes, roots, leaves, calli, and in every situation he could show that treatment with the cell wall degrading enzymes were inducing very potent defense responses in rice. And how potent they are, I'll show you in a few slides. Now this gave a puzzle. The very enzymes that are required to cause disease on rice are eliciting immune responses that can potentially stop the pathogen from causing disease on rice. So how does XOO solve this problem? And Gopalji hypothesized that XOO bacterium should be able to suppress these immune responses. So he mixed the cell wall degrading enzyme with the bacterium and infiltrated them into rice. And lo and behold, there was no immune response induced in rice. So what Gopalji was able to conclusively show was that XOO suppresses immune responses induced in rice after treatment with cell wall degrading enzymes. So the cell wall damage caused by XOO is suppressed by XOO using some other, at that time, unknown mechanism. Now, around this time, work in Arabidopsis was showing that immune responses induced by flagellin were suppressed by Pseudomonas pathogens of Arabidopsis using the bacterial type 3 secretion system. So XOO has, we knew by that time that XOO had a bacterial type 3 secretion system. And what Gopalji did was he made mutants in this type 3 secretion system of XOO, and he found that this mutant is no longer able to suppress immune responses. So what they showed was that XOO type 3 secretion system suppresses immune responses in rice caused by cell wall damage. Now the T3S, as many of you may know, is an apparatus that secretes proteins from the bacterial cell directly into plant cells. So the T3S secretes proteins directly into plant cells. Now what Gopalji also found that the type 3 secretion system of XOO is itself an inducer of rice immune responses because it continues to secrete cell wall degrading enzymes which are secreted through another secretion system. So what was clear here was that XOO secretes cell wall degrading enzymes. These incipient immune responses that are triggered by cell wall damage are suppressed by XOO using a type 3 secretion system. Now, what are these immune responses? This is a slide where this was done by Gopal. There are two rice leaves here. There's a rice leaf at the bottom into which he infiltrated a little bit of buffer, and another rice leaf in the top which has been infiltrated with either a cell wall degrading enzyme called a cellobiosidase or this type 3 secretion system defective mutant. So either the enzyme, purified enzyme, or the type 3 secretion system defective mutant. So some rice leaves were treated with either the cell wall degrading enzyme or this T3S minus mutant. Other leaves were treated with buffer. And all leaves were subsequently inoculated in the midvein, which is the infection coat for the XOO bacterium. And all of them were inoculated. And what you can see is that the rice leaves treated with buffer, there's a big lesion that is actually being produced. That is the lesion that XOO produces. In the rice leaves that had been treated with cellobiosidase or with the type 3 secretion system defective mutant, either the lesions are totally absent, as I am showing here, or they're substantially reduced. What we are actually showing here is that treatment with either a cell wall degrading enzyme or the type 3 secretion system defective mutant effectively immunizes rice against subsequent infection by the wild type bacteria. Now, there are several other assays for immune responses that Gopal and other people subsequently in the lab we use. And these are one of those hallmark assays that we use is an assay for deposition of what is called callose deposits. Callose deposition is a hallmark of the plant innate immune response, and it occurs at the point of damage in the cell wall that is caused by a microbe. What the plant does is that it strengthens that part that is under attack by deposition of callose. The cell wall is strengthened, and these callose deposits probably also contain some antimicrobial compounds that can act on the invading microbe. So if you infiltrate rice leaves with either a cellobiosidase enzyme or the type 3 secretion system defective mutant, there are a variety of callose deposits, a number of callose deposits that are produced, which are seen as these bright spots here. There's an assay using aniline blue stain, but right now here it actually shows up as bright spots on this particular leaf. A rice leaf infiltrated with the buffer doesn't have these callose deposits and has very few callose deposits. So this is one of the assays that we use to determine when an immune response is induced. So I've told you that XOO uses the type 3 secretion system to suppress immune responses caused by cell wall damage. So the next question that we had was, what are the proteins that are secreted through the type 3 secretion system of XOO? that suppress cell wall damage induced rice innate immunity. Another PhD student in my lab, Dipanvita Sinha, joined the lab at this time, and she took upon this job of identifying which are the XOO proteins that suppress cell wall damage induced rice immune responses. Luckily for us, by that time, Dr. Seiji Tsuge's lab in Japan had actually identified 16 different type 3 secretion system secreted proteins, which are called non-TAL effectors. Now, you don't have to worry too much about this particular nomenclature. Just think that there are 16 T3S secreted proteins that XOO secretes into rice. What Dipanvita did was she tested each one of these proteins in transient transfer assays. She treats with the cell wall degrading enzymes and transiently also expresses one of these 16 proteins. So she wanted to find out whether treatment with the expression of this particular ZOP, one of these T3S secreted proteins, would that suppress cell wall damage induced immune responses. And what Dipanvita found was that out of these 16 proteins that she was testing, 4 of these proteins were able to suppress immune responses in rice that are triggered by cell wall damage. These proteins are what are called Xanthomonas outer proteins, or ZOPs, and there are 16 of them. They're different nomenclatures, and these are proteins called ZOP N, Q, X, and Z. Again, the nomenclature is not important. Just take it that there are 4 ZOPs that suppress cell wall damage induced immune responses in rice. Now, this is an example of how we do this particular assay. So into one rice leaf, we infiltrate the cell wall degrading enzyme, and there are a lot of callose deposits. Into the same leaf, along with the cell wall degrading enzyme, we infiltrate an agrobacterial construct that encodes ZOPQ in it in a plant on a plant inducible promoter. And you can actually see that expression of ZOPQ suppresses these callose deposits. Now, this happens not only in rice, but also in Arabidopsis. So by this kind of an assay, Dipanvita was able to show that 4 ZOPs can suppress cell wall degrading enzyme induced immune responses in rice. The next thing Dipanvita did was, okay, each one of these proteins can suppress immune responses, but what happens if you knock them out? So she started making single, double, triple, and finally she ended up with a quadruple mutant that is defective in all 4 ZOPs. So in one rice leaf here, she infiltrated water. A few callose deposits can be found. Into another rice leaf, she infiltrated the wild type XOO bacterium. There are a few callose deposits. If she infiltrates the ZOP N, Q, X, and Z quadruple mutant, again, there are lots of callose deposits that are produced. The type 3 secretion system defective mutant also induces callose deposits. Now, what is interesting here is that if she knocked out one gene, one of these ZOPs, that didn't affect innate immunity suppression. 2, that a double mutant can also suppress innate immunity. Triple mutant can also suppress innate immunity, but the quadruple mutant cannot suppress innate immunity. It itself becomes an inducer of innate immune responses in rice, again because it continues to secrete the cell wall degrading enzymes. So what they showed was that the ZOP N, Q, X, and Z proteins function redundantly in suppression of post-innate immunity. So the next question was, how do these ZOPs suppress rice immune responses? And this was taken up by another PhD student in the lab. Her name is Sohini Deb. And Sohini Deb and another postdoc called Mahesh were actually working on these proteins, trying to find out these 4 ZOPs, ZOP N, Q, X, and Z, what kind of biochemical activities they have, what are the other features in their sequence that might actually give us clues to how they suppress immune responses. What we were able to show for ZOPQ was that the biochemical activity of ZOPQ, again, what the biochemical activity is not pertinent for today's talk, but we were able to show that the biochemical activity of ZOPQ was not needed for suppression of innate immunity. We couldn't predict a biochemical activity for either ZOP N, X, or Z, but for ZOP Q, X, and Z, we could find motifs that are actually labeled as 1433 by protein binding motifs. Now, as you all know, the 1433 proteins are key adapters in eukaryotic signal transduction cascades. And this was interesting for us because by then several Pseudomonas and Xanthomonas pathogens had been shown to modulate host immune responses in other crops by binding to 1433 proteins. So what Sohini Dev did was, rice has eight 1433 proteins, and she tested each one of them against ZOP Q, X, and Z to see if they would actually, if these ZOPs would actually bind to rice 1433 proteins. And it turns out that each of these ZOPs, Q, X, and Z, bind to unique rice 1433 proteins. For example, ZOP Q actually binds to GF14F and GF14G. ZOP X binds to GF14D and GF14E, while ZOP Z binds to GF14B. Now, the 1433 proteins are called GFs because they're labeled as general regulatory factors because they're so important in regulation in eukaryotes. So what they showed is that by yeast 2 hybrid assays and DIFC, Sohini was able to show that each of these ZOPs bind to a distinct set of 1433 proteins in rice. What Sohini was also able to show was that if she mutated, she made a point mutation, she makes a point mutation in the 1433 binding motif of either ZOP Q, X, or Z, these proteins lose the ability to bind to their target rice 1433 proteins. Not only that, these mutations in the ZOPs that are in the 1433 binding sites, they are also not able to suppress host innate immune responses. If you're interested, we can discuss how we did it, but take it from me right now that that's what we actually found. What this indicated was that the ZOPs of XOO are suppressing rice immune responses through interaction with 1433 proteins. And it appears that each of these ZOPs is binding to a different 1433, different set of 1433 proteins. So the next question that came up was, and this is something that we are still addressing in the lab, what might be the role of these rice 1433 proteins in innate immunity? As I told you, there are 5 different rice 1433 proteins to which these ZOPs found. What has already been shown is Professor Jan Leach's lab at Colorado State University has shown that one of these 1433 proteins called GF14E is a negative regulator of rice immune responses. Now, what we have done is that we have transiently overexpressed the other 4 1433 proteins to which the ZOPs bind. And what we find by a variety of assays, that if we transiently overexpress any one of these 4 proteins, we trigger immune responses in rice. We are now trying to follow up these transient assays by generating stably transformed rice lines that overexpress these 1433 proteins in an inducible manner and trying to see if those would also trigger immune responses. But what the preliminary data suggests is that these other 4 1433 proteins to which the ZOP bind, they appear to be positive regulators of rice immune responses. Now, what all of this shows is that the ZOP Q, X, and Z suppress rice immune responses by interaction with 1433 proteins that are modulators of rice immune responses. So what we have also been doing in parallel is to try to understand what are the rice functions that are involved in induction of cell wall degrading enzyme induced innate immunity. Due to paucity of time, I'll quickly summarize this data. What we initially did was we treated rice cells with cell wall degrading enzymes, did a transcriptome analysis at early and late stages after treatment with the cell wall degrading enzyme. And at the early stages, we looked for receptor kinases whose expression might be upregulated following cell wall damage, because there is evidence that the receptor kinases are actually the ones that are involved in perception of pathogen-triggered immune responses in plants. Now, what we found was that there was a wall-associated receptor kinase called OS-VACL21 that's triggered within an hour, whose expression is triggered within an hour or hour and a half of treatment with cell wall degrading enzyme. At a later stage after treatment with cell wall degrading enzyme, a particular transcription factor called OS-RERJ1, which is a helix loop helix transcription factor, it's very highly upregulated at the 12-hour time point after treatment with cell wall degrading enzyme. So along with some other proteins, these two are those, along with some other differentially expressed proteins, we've been characterizing OS VACL21 and OS REG1. And we have most advanced in our understanding of these two proteins and how they might function in plant innate immunity. What we find is that overexpression of either one of these two genes in rice or in Arabidopsis results in induction of immune responses. Now, knockdown of expression of OS VACL21 in rice using RNAi or a mutant in OS REG1 that we isolated by screening through a tilling population. Mutants that are knockdown or knockdown of OS VACL21 or mutants of OS REG1, they result in reduced immune responses in response to cell wall damage. So what it suggests is that OS VACL21 and REG1 are important players in elaboration of cell wall damage induced immune responses in rice. Now I'm just showing you a very simplified scheme for how we think cell wall damage in innate immune responses are induced in rice. We think that there's a receptor in the plasma membrane, and that receptor could be OS VACL21. So OS VACL21 could be either the receptor or a co-receptor, and we think that this binds to an elicitor that is released upon cell wall damage caused by the cell wall degrading enzyme and triggers a signal transduction cascade. And we think these multiple 1433 proteins to which the ZOPs bind, that they are involved somewhere in this signal transduction cascade. So several of these steps, some of these 1433 proteins are working. The net sum of activation of receptor kinase and multiple signal transduction cascade mediated by these 1433 proteins and yet other proteins that we have not yet discovered results in activation of transcription factors. One of them is OS REG1. I'm saying OS REG1 is one of them because we think other transcription factors are also involved. But activation of these transcription factors results in induction of genes involved in the defense, and that results in a defense response, and that results in resistance. What we think the ZOPs are doing, and again, this is a simplified scheme for suppression of innate immunity, we think that the elicitor may be still binding to the receptor, but the signal transduction cascade is actually interrupted by these ZOPs. So even if there is activation of the receptor, which we don't know that it is there, but we presume that the receptor might still get activated, but because these ZOPs are around, they stop the signal transduction cascade. There is no activation of transcription factors such as OS REG1. There's no defense response, and that's what results in susceptibility. Now, just I want to summarize, going back to the first slide that I had, one of the early slides that I showed. What we had found was that secretion of cell wall degrading enzymes by XOO results in triggering of defense responses in rice, and this is the equivalent of pathogen triggered immunity. XOO is able to suppress these using effectors that it secretes into rice using the type 3 secretion system. So XOO suppresses PTI. Now, what I have not shown you is that some of these ZOPs, they actually interact with each other. And when they interact with each other, there is actually, and we don't know how this works, there is again a triggering of defense responses in rice. There is what is called effector triggered immunity. So the very effectors that suppress PTI, they are triggering an immune response. What is interesting is that XOOs has several other T3S secreted proteins, some other ZOPs that we have also identified, and these suppress ETI. So the net result is that XOO is able to cause disease in rice, but it is able to do it because it is able to suppress both layers of the plant innate immune response. It is able to suppress pathogen triggered immunity, and it's also able to suppress effector triggered immunity. So what next? We think that the key issue that we have to address is to try and find out what is the exact role of each of these 1433 proteins during elicitation of cell wall damage induced rice immune responses. So we are working on this aspect. Now the next point is, along with these 1433 proteins, what might be the other proteins that are involved in linking up activation of the receptor to activation of the transcription factor? We know the 1433 proteins are there, but there must be others. We are trying to get those also. And the big question is, how does interaction with the 1433 proteins enable these ZOPs to suppress rice innate immunity? What we've just started doing, and this is something that I didn't present, we have shown that two of these ZOPs, which are called ZOPQ and ZOPX, Individually, if you introduce them into rice, they suppress innate immunity. But if you introduce them together, they interact with each other, they're able to bind to each other, and somehow when you put both of them together, that triggers immune responses in rice. And there are other ZOPs that suppress innate immunity. So we want to try and find out how ZOPQ and ZOPX trigger effector triggered immune responses in rice, and how do these other XOZOPs suppress them. And this is work done by a lot of people at CCMB and at NIPGR, and I'm not going through each of their names because there are a lot of talented PhD students and postdocs who've been involved in this work. And I also work on other aspects of this XO rice interaction, where we try to develop novel rice varieties that are resistant to the bacterium. And I have collaborators at CCMB and at NIPGR, as well as at the Indian Institute of Rice Research, which is one of the leading rice research institutes in India. We also collaborate with D.O. Mishra at Biocrop Science on looking at pathogen diversity within the country and how that can help us choose what kind of rice varieties need to be developed to resist XOO. Now, thank you for your time, and thank you for listening to me. I look forward to hearing your feedback. Thanks.
Great, thank you. Thank you, sir. Thank you, Dr. Sonti. That was a very good talk, especially coming with 20 years, the temporal scan of all the work that has been done in your lab. And you kept saying you don't name names, but it was wonderful to see you remembering every student from the beginning who has done the work and contributed to the work that you've done and the results in your lab. So that's really wonderful. And while we are waiting for a couple of questions to be typed in the chat box, oh, well, there are a few questions in the chat box, but because I get to decide, I'd like to ask you first. I'm so interested in the fact that you got the 14.3.3s and specificity in 14.3.3s, because usually these are highly conserved domains, the 14.3.3s, and really tiny domains, all alpha helices mostly. And quite a lot of these 14.3.3s have been, usually the moment you find 14.3.3s are around and they're working together, that happens in the mad cow disease also, for example. But in your case, you have noticed a specificity, which is not what one expects from very highly conserved domains like the 14.3.3. And you found the ZOPs are specifically recognizing the 14.3.3s. So I'm very interested in whether you have taken a look at whether the ZOP protein structures are known, because 14.3.3 is known. So is there a way we could look at what it is, where the specificity is between them? Or do you think that is not possible because of the ZOP structure not being known? Basically, what is your take on why the specificity?
It's a good question, Gitanjali. We are really not sure what determines the specificity. But what we have actually found is that there are certain residues in the ZOPs that if we mutate, they are not able to bind to the 14.3.3 proteins. And part of it was through modeling, and part of it was through some deductive logic that we made looking at the structure. Okay. So we have particularly done that for ZOPQ, whose structure is known. So yes, there are residues that we can identify, but what actually leads to the specificity, how one ZOP will bind to two 14.3.3s, another ZOP will bind to another set of 14.3, we really don't know, to tell you the truth. There's a logic there that we have to figure out what is going on.
There's a code there, surely, and that needs figuring out. But it's very interesting to hear that coming from you. I will now take the questions that other listeners have typed out in the chat box. There's a question from Sabine Zimmerman, and Sabine is asking, what is it that allows the plant to distinguish between pathogen and the symbiont?
Yes, that's a good question. One of the things that we are thinking about is that it may be the kind of damage that is done to the cell wall. A pathogen may actually secrete more and a larger variety of cell wall degrading enzymes than a symbiont might actually do. Because all of them probably have at some stage or another some amount of damage they create to the cell wall. So that's at one level that it might occur. At another level, it may be that the symbiont has certain other functions that it is able to modulate the plant immune response so that the plant doesn't recognize it as an enemy, but it recognizes it as a friend. But it's a very important question, and we don't really know what the answer is, to tell you the truth.
I hope that answers your cues, Sabine. There's a question from Garima Pal, who's currently a PhD student at the RCB, and she's asking about TAL effectors. She's saying, since they're highly conserved, can you distinguish these TAI effectors from each other?
Yeah. Okay. Just I wanted to, I mean, this is something that I didn't cover. There are two classes of proteins that are secreted by bacterial plant pathogens, particularly the Xanthomonas, using the type 3 secretion system. There are the TAL effectors, which are called the transcriptional activator-like effectors. They are actually proteins that are secreted by the bacterium. They go into rice. They act as transcription factors to upregulate post-susceptibility factors. So those are the TAL effectors. What we are working on are the non-TAL effectors. So there's another set of proteins that XOO secretes that go into rice, and these are very diverse proteins. They're not really related to each other, and each one of them is probably doing a different function, okay, other than binding to 14-3-3s. Okay, so the non-TAL effectors are very easy to distinguish one from the other.
I see. Okay. There's this question.
They're quite different from each other. It's the TAL effectors that are very related to each other.
Okay.
So you
okay.
can distinguish the non-TAL effectors quite readily.
Thank you, Dr. Sonti. There's one question from Raman Sundaram, and he's asking, is there any scope for doing genome editing of the 14-3-3 protein encoding genes to suppress the nexus between ZOP and EDI suppression? What is your take on that?
Yeah, no, that's a good point. We are actually trying to take the negative regulator and trying to see, which is GF14E, which we are actually trying to do genome editing. It's a negative regulator, and our idea is that if we make a genome edited line that is knocked out for it, then maybe we can actually, have immune response that is triggered, and maybe those plants will be more tolerant to the bacterial, either bacterial or fungal infection. What Dr. Jan Leed's lab has shown is that RNAi knockdown of GF14E confers tolerance against XOO and against fungal pathogen. So yeah, it's possible. We are not sure yet how we can actually do that for the positive regulators, but I think the path is straightforward for the negative regulator, the GF4, the 14-3-3 protein that serves as the negative regulator.
Thank you. And I will take one last question in the interest of time. The question is from Zahur Mir, and he's working on Alternaria brassica interactions. And they observed that the Brassica juncea is susceptible compared to another Camelina saliva to Alternaria infections, and they did proteomics analysis. They did not get any positive results, and now they're doing RNA-seq, and it is still happening. So they're saying, to our knowledge, we did some, we also did some real-time expression analysis, and we find that in Brassica, the early expression of SA-mediated pathway genes, but not JA pathway genes, ricartinase and defensins is going on. What could be the reason? Okay, I feel that this should be taken on directly with you at another point, but if you want to take up this question, Dr. Sonti, please feel free.
See, basically, it's non-host resistance. Brassica is a host for Alternaria, while I think Camelina is actually a non-host. So how non-host resistance works? Is it because the suppressors, there are, I mean, this is just my my take, coming as someone who works on host immune responses. So when you have resistance, it's either because pathogen-triggered immunity is not being suppressed, or effectors that suppress pathogen-triggered immunity are actually triggering, the effectors that suppress PTI are triggering ETI. So I mean, this is what I would actually say, that it's probably at the level of actually an inability to suppress host innate immunity in the non-host plant, but in the host plant, it's able to suppress innate immunity. That's just my thing, but I'm not a specialist in this area, so I can't tell. I look at it only from the prism of immune responses. If it suppresses the immune responses, then it is able to cause disease. If it can't suppress immune responses, it can't cause disease. It's a rather simplistic statement, and I'm maybe wrong, but that's just something that I can suggest there right now. And how do you tackle whether camelina is able to Alternaria is not able to suppress immune responses induced in camelina? You have to look at the readouts of immune responses in plants upon treatment with Alternaria and see if those actually are suppressed. They are not actually induced in camelina, but they are induced in brassica. So just offhand, what I can tell you.
There is a related question that I therefore want to take up now. The question is by Kamali S. And they're saying, is there any evidence that the innate immunity caused by cell wall degrading enzymes may be converted to plant memory of some kind and, carried on temporarily or long term?
Sure. No, but we have not looked at it, Gitanjali. Okay, we've not really looked at memory and whether, how long the immune response state persists. We have not actually done that. No.
Have you looked at key regulators like LNC-RNA in regulating CWEs? This is a question from Neha Pasrekar.
No, we have not done that. No, we have not looked at LNC-RNAs. We have not looked at microRNAs. We've just been looking so far at the proteins that are involved in this process.
Well, I thank you once again, and I would like to, at this point, give the charge back over to Katie. Katie, I know that you've been showing us lots of slides about what's happening at Plantae. Please feel free to take this from here. And I thank both the panelists, of course, both the speakers today.
Yes, and thank you, Gita, for doing such a great job of moderation. For me, it's morning, so this was a spectacular way to start the day. I really appreciate y'all being here. Thank you all for joining live. I've been kind of scrolling through the names, and I know a lot of you oftentimes stay up late. So thank you for joining and participating live with all the questions. We'll see you at the next event. Bye, everyone.
Bye, and thank you all.
Thank you all.